Good morning. My name is Duarte Pinhatelli. I am from Porto, Portugal, and I have devoted myself to study polycystic ovary syndrome for a long time. I'm here to talk to you about the genetic causes of polycystic ovary syndrome. Hello, good morning. My name is Duarte Pinhatelli, and I'm from Porto in Portugal. And I've been devoting myself to study polycystic ovary syndrome for a long time, and I'm going to talk to you about possible genetic causes of PCOS. Polycystic ovary syndrome is a common endocrine disorder in females, especially in the reproductive age. The worldwide prevalence of polycystic ovary syndrome is estimated to be 5 to 10%. Although the exact etiology is unclear, PCOS is currently thought to emerge from a complex interaction of genetic and environmental traits. The exact cause of aberrant follicle development is unknown, but follicle arrest is associated with elevated serum concentration of LH, anti-Mullerian hormone, insulin, androgens, and the relative deficiency of FSH. Recent genome-wide association studies have found susceptibility lossy close to FSH receptor and FSH beta polypeptide gene and the LHCG receptor, implicating gonadotropins and their receptors in the pathogenesis of PCOS. Preceding the clinical and reproductive and metabolic aspects that we see in our patients, a lot of changes occur, hormonal changes exacerbated by obesity, and in the genesis of these hormonal changes, there are genetic alterations and lifestyle modifications that contributed to them. Also, epigenetic alterations may be part of this puzzle. What we know is that PCOS is strongly a familial syndrome. Concerning the genetic basis, we are not so sure because many results are hard to replicate and there is failure to maintain linkage with PCOS phenotypes. Can inheritance be modified by environmental factors? Is there an influence of reprogramming of genes in utero? PCOS is a polygenic and multifactorial syndromic disorder involving genes of ovarian and adrenal steroidogenesis, genes involved in steroid hormone effects, genes involved in gonadotropin action and regulation, and genes involved in insulin action and secretion, as well as other genes. So let's start talking about genes involved in ovarian and adrenal steroidogenesis, including the genes of the enzymes CYP19, CYP17, CYP21, and CYP11 alpha. CYP11 alpha encodes an enzyme that is essential for the conversion of cholesterol to progesterone, a rate-limiting step in the conversion of cholesterol to the other steroids later on. In 1997, Garani was the first to report polymorphisms and genetic variants associated with infertility in women. Two other studies replicated the finding and reported these mutations as being associated with PCOS. However, not all the studies confirmed this later on. CYP17 is an enzyme much more related to the production of androgens, and androgens are reported to be increased in PCOS patients, and so this enzyme looks and is an essential step of the pathogenesis of PCOS. This regulation of this enzyme was demonstrated to be causal of polycystic ovary syndrome since 1990. Expression of CYP17 in polycystic ovary syndrome TECA cells was demonstrated to be increased, and the promoter activity was demonstrated to be 4 times increased in those cells. Carey and other collaborators had previously reported a polymorphism in the promoter region that was associated with PCOS. P450 aromatase is responsible for the conversion of androgens to estrogens. Lower activity of aromatase has been reported in both obese and lean women with PCOS. No mutation was found in P450 aromatase gene, but this does not preclude the role of aromatase deficiency in the pathogenesis of PCOS. Hello, my name is Duarte Pignatelli, and I have been devoted to studying polycystic ovary syndrome for a long time, and I'm going to present my analysis of the genetic causes of PCOS. Good morning, my name is Duarte Pignatelli and I'm from Porto in Portugal. I've been devoting myself to study polycystic ovary syndrome for a long time, and I'm going to present you some analysis of the genetic causes of polycystic ovary syndrome. And now let's move to the genes involved in steroid hormone effects involving the androgen receptor and the globulin that transports sex hormones. The androgen receptor gene is located in the long arm of chromosome X. Mutations and structural disruption of the gene have been reported to cause PCOS. X chromosome inactivation is sufficient to lead to an elevated androgen hormone production, resulting in PCOS as well. Sex hormone binding globulin is a glycoprotein mainly produced in the liver, which regulates bioavailability of sex steroid hormone. Single nucleotide polymorphisms in the SHBG gene was described to be significantly associated with PCOS in numerous studies. And now let's talk about genes involved in gonadotropin action and regulation. And we will now continue with genes involved in gonadotropin action and regulation. Both LH and the dysfunction of its receptor, LH receptor, are frequently reported as a cause of PCOS. A high level of LH enhances the production of androgen, but especially there seems to be a premature activation of granulosa cells that cause follicle development arrest. An association between polymorphisms of LH receptor and PCOS has been frequently reported, indicating that variants of this gene may affect the pathways involved in the syndrome. Anti-Mullerian hormone is a member of the TGF-beta superfamily, and it's responsible for the Mullerian duct regression in male embryos. Its maximal expression occurs in the females in preantral and small antral follicles. Expression disappears in maturing preovulatory follicles, as you can see in this image. In females, anti-Mullerian hormone is a regulator of folliculogenesis. AMH decreases the androgen biosynthesis by inhibiting CYP17 activity. Several genetic variants were identified in women with PCOS. These mutations lead to the PCOS phenotype by abolishing the transcriptional inhibition of CYP17 that AMH used to do. FSH receptor gene encodes for a G protein coupled receptor which is required for gonad development. Mutations of the gene disrupt the structural protein, thus causing an imbalance of the hormone, and this hormonal imbalance may cause PCOS. SNPs in the FSH receptor gene, similarly to those in FSH beta subunit as well, might also contribute to PCOS by altering its phenotype. And what about the genes involved in insulin action and secretion? Insulin resistance and the hyperinsulinemia that results from it are very much connected to PCOS pathophysiology and pathogenesis, namely at the level of the pituitary, where they cause alterations in LH pulse and amplitude, and at the ovary and adrenals, where they increase the CYP17 activity. Both of these alterations leading to increase of androgens at the level of the ovary and the adrenals, and contributing to the development of PCOS. Insulin plays a significant role in the production of androgens through its receptors that are present in TECA cells and adrenal and many other organs. The mini-satellite variable number of tandem repeats region of the chromosome upstream of the insulin gene regulates its expression, and several studies have looked for association between these VNTRs with PCOS. Recent meta-analysis confirmed the strong association of insulin VNTR's polymorphism with PCOS risk only in an ovulatory woman but not in overall women with PCOS, which may explain some contradictory results that were obtained. And here we see a summary of the studies of linkage and association of insulin secretion and action aspects with PCOS. Involving insulin receptor, the VNTRs, and the insulin receptor substrate 1 and 2, as well as calpain 10, PPR gamma, and PPP1R3. The IRS1 and 2 are downstream effectors of the insulin receptor. Several studies have been performed to find out the association between the genes of IRS1 and 2 and PCOS. And although there are contradictory aspects, maybe the solution for this contradiction is that the association between mutations of IRS1, for instance, are related to metabolic features of women with PCOS and not the PCOS itself or completely. In what concerns calpain 10, this is an important molecule in beta cell function. It is likely to be determinant of fuel sensing and insulin exocytosis, with action in the mitochondria and plasma membrane, respectively. Several polymorphisms in the calpain 10 gene were associated with increased PCOS risk. And now let's have a look at other genes like 5-alpha reductase gene and FTO. The fat mass obesity, also known as FTO gene, is associated with obesity and type 2 diabetes. There might be a direct association between FTO variants and the PCOS risk independently of BMI, as was demonstrated in several studies. The case of 5-alpha reductase relation to PCOS is even more curious. 5-alpha reductase is responsible for the conversion of testosterone to the hydrotestosterone and was demonstrated to be enhanced in women with PCOS. There were many studies testing the susceptibility of these genes in PCOS, and it was reported that a variant of 5-alpha reductase, 5A2, was associated with protection to PCOS, while variants of 5-alpha reductase A1 were associated with risk of hirsutism and thus risk of PCOS. The DEND1A gene was identified as a potential risk marker for PCOS in GWAS studies. A variant of DEND1A is overexpressed in PCOS TECA cells compared to normal TECA cells. Taking all data into consideration, DEND1A variant 2 is potentially one of the mechanisms involved in the intrinsic abnormality of ovarian TECA cells in PCOS. DEND1A encodes a protein associated with clathrin-coated pits where cell surface receptors reside. This protein is located in the cytoplasm and in the nuclei of TECA cells, and as you can see here, it's much more expressed in PCOS than in a normal follicle. And here you can see the Western blot analysis of DEND1A in normal and PCOS TECA cells, clearly demonstrating that it has a much higher expression in PCOS than in normal follicles. And in this last slide, I present you the results of a very recently published paper in GCEM concerning the gene expression in granulosa cells from small antral follicles from women with or without polycystic ovaries. And you can see that there is increased LH receptors in the subpopulation of follicles, and at the same time reduced expression of FSH receptor, CYP11A1, and androgen receptors. These changes, in particularly this aberrant expression of LH receptor function, but also reduced FSH receptor expression, are key factors leading to the premature arrest of antral follicle growth and so to the pathogenesis of PCOS. And this concludes my presentation about genes and PCOS, hoping that I demonstrated that there are lots of pathways being studied and lots of genes that may have influence, but we still have a lot more to understand and to know what the real influence of genes in the pathogenesis of PCOS. Thank you.